Okay, very good morning, my dear friends. Welcome to today's session. CPGET 2025 free online coaching for all competitive exams, especially botany, plant sciences, and plant biotechnology, and other competitive exams. Today's our eminent resource person, Dr. THIRUNAHARI UGANDHAR sir, Associate Professor, Department of Botany, Kakatiya Government College, Autonomous, Hanmakonda. Sir is going to explain about today in the molecular biology, one of the most important topic, that is chromosome structure and morphology, chromosome components of the chromosome, and the discovery and the history of the chromosome, and the functions of the chromosome. Sir is going to explain. So, my dear friends, utilize this wonderful opportunity. Sir, please welcome and start your session, sir. Please welcome. Yugandhar sir, start చేయండి.
Okay, sir. So start that. Today our session, second session, topic chromosome morphology. So what is a chromosome? What are the structure of the morphology, functions, and how to made up of the chromosomes? So these are all is today we are discussing that. So the chromosomes which are present in the nucleoplasm in the form of the chromatids. The chromatids is which jointed part of centromere. Then either side of the centromere, two chromatids are present. Each chromatid which is made up of several number of supercoiled structures is called a nucleosomes. So these nucleosomes are ending part is also called a DNA. The DNA which is important of genetical material for living organism. Any live which responds to the only genetical part is called DNA and protein. Both are important. The DNA, protein, and RNA. That is also important particles for the formation of the life. The life which is, so what are the chromosome? The chromosomes are thread-like structures in the nucleus. Was a first reported Edward Strasburger, made of DNA coiled around stone proteins. Support a structure by stones. The stones means lysine and arginine. Rich part is histoproteins. These histoproteins are made up of the chromosome. The chromosome name, the chromosome structure was first described by Strasburger in 1815. The term chromosome coined by Waldeyer in 1888, visible as rod-shaped bodies during the metaphase. Only chromosome which can observe under the microscope during the metaphase, not prophase and anaphase and telophase. Only metaphase is a suitable stage for identification and observing of chromosomes on the mitosis. This was a first reported, that is called Edward John Strasberger, was a kind of the not name the kind the chromosome, only description. So the chromosome function is which can takes place in DNA or protein. That is also confused. But the Waldeyer scientist was a kind of the name of the chromosome. This is also this scientist was a Waldeyer scientist. So this is Waldeyer. Heinrich Wellman Gottert Waldeyer Hirsch, 1836 to 1921, was a German anatomist, recognized as one of the forefather of anatomical science. Then Waldeyer's Fisca and Waldeyer's Ring are two of his more well-known eponyms. Waldeyer introduced the term chromosomes and neuron to medical terminology. So this the conventionally the published incorrect and how to dish of views on the anatomical differentiation of African and female neuroanatomy. So this is a first scientist who was a reported, not reported means kind name. The reporting or description is also called Strasburger. So this is also Edward Jenner. The what are the important of the chromosomes in the nucleus of each cell? The DNA molecule is packaged into thread-like structure called the chromosome. That is a complete or correct definition. In the nucleus, each cell means somatic cell, also reproductive cells. The somatic cells are vegetative cells, which are located on the vegetative parts or vegetative body of the plants and animals. The DNA molecule is packed into the thread-like structure, then called the, when you say that chromosomes. The chromosomes are the nuclear components of special organization, which individually function of inheritance. Inheritance means the parental characters are carries to the daughter generation by chromosomes. These functions are capable of self-reproduction, means self-replicated to two DNAs. The one DNA is a multiple two DNA. In the same way, the one DNA, two DNA formation, the chromosome is also same. One chromosome to two chromosome is called a karyokinesis, means division of the nucleus, division of the cytoplasm. Both are responsible to the cell division or mitosis. Mitosis, self-reproduction, and plays a vital role in the hereditary mutation, variation, and evolutionary development. The majority, then four type of the function as the first point is also called, it is also heredity. Heredity means it can carry inheritance characters and parental characters from generation to generation is called a heredity. Then mutation means suddenly changes to the new characters from the parental to daughter generation is called a mutation. This mutation word was first reported Hugo de Vries scientist was the first scientist to reporting the mutation. The next one in the variations. The variation is a not suddenly changes, just few generation after later it can changes of the new identical characters, and also evolutionary development of the species. Evolutionary means the origin. Origin means then the primitive body or primitive organisms are they develop into the advanced types. Few years later, it can be development. That is called origin. Then that origin is must take space in the chromosome structure, chromosome activity, and also called nucleotide arrangements. These nucleotide arrangements is which is also different in the different type of the organisms. That is called a evolutionary development of the species. Each chromosome is made up of DNA tightly coiled in many times around proteins called the histones and support the structure. Histoproteins and non-histoproteins both are important components of the chromosomes. Then chromosomes were first described by Strasburg in 1815. You know that the term chromosome was used by the Walter in 1888. So they appear as a rod-shaped line. Rod shape means linear model. Only then the bacteria and also yeast and other are microorganisms are the chromosomes are identical material is a circular or spherical in manner. But in human beings and all plants, the DNA structure also called the DNA located part is called a chromosome is the linear model. The chromosome was then each chromosome typically has one centromere and one or two arms. So that is a project from the centromeres. Then conjunction, then connections between the two chromatid between parties called centromere. Centromere either kinetophore is present. That is a part of the character. So this was a Edward Strasburger was a first pioneer German plant cytologist known for the clarifying a nuclear division in plants. Nuclear division means karyokinesis, cytokinesis and karyokinesis, which are responsible to the one cell division, which which is a performing the one cell to develop the two cells. He is a accurately described the embryonic sac in gymnosperms and angiosperms and discovered a double fertilization in also flowering plants also. The Strasburger is also pioneer of the chromosome and also called a double fertilization, means the male nuclei and female nuclei to form a zygote, that is also syngamy. Then other male nucleus fuse with a diploid nucleus to form a triploid endosperm nucleus, that is also triplogamy and also called a monogamy or digamy. Triplogamy, digamy, both are called triplo or double fertilization. The double fertilization process was reported in first time by Strasburger scientist. In his 1876 was a Uber Jellibil and a Jellium is outlined the principles of mitosis, later stating that new nuclei form only from existing ones. The Strasburg also introduced the term cytoplasm and also nucleoplasm, both cytoplasm, nucleoplasm, and also called the double fertilization and also chromosome identification. These are all events. All pioneer events are reported by Strasburger. He proved that heredity is mainly carried by the nucleus. Which nucleus? Somatic nucleus to carry the somatic character. Gametic nucleus also carrying to one generation to other generation, that is called a sperms, and also called a ova, also egg, which are responsible to the carry the inheritance character of a reparative and also gametic and a vegetative structure are the vegetative character features are carries from generation to generation by vegetative cells of the chromosomes. The carried by the nucleus and stabilized the occurrences of the meiosis in angiosomes or germ cells. The meiosis, which meiosis, what occurs? The first type of the division in the formation of the life is called a meiosis. Without meiosis, do not formation of the life on the earth. That is, the meiosis is a major important part that can reduce the chromosome number and fuse to the diploid zygote formation. The zygote is also structurally also called different to the vegetative structures of the organisms. So in nucleus and stabilized the occurrences of the meiosis in angiosomes of the germ cells. That is also, so yes, it is also completed, but that is also what are the important of the character features. So these are all is also the nucleic acids and the chromosome structures. The chromosomes in a different staining process. During the staining process, the color of the chromosomes and the structure of the chromosomes are easily identified under the electron microscopes. So this is also. Then what are the character features of the structure of the chromosomes? The structurally each chromosome is a differentiation to the three parts only. First outer part is covering the thread-like structure is called a pellicle. Then pellicle inside is juice or insected of the fluid-like structure is called a matrix. Then chromonema, thread-like structure is called a chromonema. The three parts which can divide it into the every chromosome or reparative chromosome, also called somatic chromosomes. In the pellicle, it is the outer envelope around the substance of the chromosome. It is a very thin and is a form to the achromatic substances. Acromatic and achromatic, the two types of the substances are present in the chromosomes. So this is also called a matrix. It is outside of the layer is also pellicle, black color part. Then white color part is called a matrix. In the white color part inside, thread-like parts are present. These threads is also called a important of the and a chromonema filaments. Each chromonema filaments which are responsible to the in futurely or developingly to the a large number of the DNA molecules are present. So then each pellicle is also complete is a very thin and strand. Thin and is a formed acromatic structure matrix. It is the ground substances of the chromosome which contains the chromonemata filaments. This chromosomal filament, it also forms the non-genital material. Genital material, non-genital material. The matrix, the matrix is also liquid-like structures. It is a non-genital material part. Then pellicle is also called non-genital material. Only chromonemata as genital material of the chromosomes. Then it is the chromonemata embedded in the matrix. The matrix is a liquid. Then chromonemata is also solid type and a thread-like structure, also called a curved of the nature. Of each chromoneme are two identical spirally coiled threads called the chromonemata. Then also called a thread legs. The two identical copies means either side of the centromeres. The two chromonemata are also tightly coiled together so that they appear as a single thread of about 800 angstrom thickness. A large number of the chromonema filaments are come close to tightly packed. That is called chromonemata. Chromonema means singular, chromonemata means plural. That is important. But it is in eukaryotic cells, the chromosomes are composed of the single molecule with many copies of the 5 types of the histones are there here. 5 types of the histones which can important for the components of the chromosomes. The histones are proteins molecules and are in a rich in lysine and arginine are residues. So they are a positively charged. Hence, they bind tightly to the negatively charged phosphates in the DNA sequences. Then here is also PGT-like structures of the substances which can made by the chromosomes. A small number of the non-histoproteins are there. Histoproteins are non-histoproteins. The non-histoproteins are also present. These are a mostly transcription. Transcription means it can be genetical material or genetical characters of the parent generation to daughter generation are transcripted by RNA. This RNA is major role to transcripted of inheritance characters from the parent to daughter. That is called a important role in the RNA. So during the cell's life cycle, chromosomes are elongated and cannot be observed under the microscope. It can only observe under the microscopes and yes, are synthetic phase of the mitotic cell cycle, the chromosomes are duplicated. Duplicated means just one is a short and also thick type of the chromosomes. At the beginning of mitosis, the chromosomes are a duplicate and they begin to the condense into the short structure which can be stained. The starch structure which stained means it is a coloring parts that can saffron. That is also one type of the substances which are responsible to the stain. So they stained and observed easily under the light microscopes. These duplicated condensed chromosomes are known as dyads. It is as dyads. Dyads means it is also duplicated parts of the chromosome which is appeared on the S synthetic phase. S1, S2, G. These 3 type of the stages which observed into that is also special structure of the chromosome is also dyads. So the duplicated chromosomes are held together at the region of centromeres. Centromere region part, it can having a duplicated part. The centromere is means center part of the chromosomes, either side of 2 chromatids. The centromere in women are made up of about 1 to 10 million base pairs of the DNA. Base pairs means nucleotides. The nucleotides and nucleosides, which are a base pairs. The DNA of the centromere is a mostly repetitive short sequence of DNA. The sequence are repeated over in a tandem arrays. So the attached duplicated chromosomes are commonly called the sister chromatids. then duplicated chromatids and original chromatid. Then duplicated chromatid parts, attaching part is also called a sister chromatids. These sister chromatid parts, then either side or between or below kinetochore are the attachment point for spindle fibrous, which helps to the pull a part of the sister chromatids as the mitosis process proceeds to the anaphase phase. During the anaphase phase, so this anaphase is also referred to the chromosome structure, which is also just changes in anaphase. The mitosis process proceeds to the anaphase stage. The kinetochores are the complex of about 80 different proteins are present. The shorter arm of the two arms of the chromosomes extending from the centromere is called the P arm. P means that is also arm. Arm is one type of the part of the chromosome. And the longer arm is known as a Q. Shorter arm is called a P. This is also two type of the arms are present here. So this is also known that here is also telomeres, then short arm and also called a long arm. Here the two type of the arms are here. The short arm, short, but here is the long arm. Long arm. This center part is called centromere. But here yellow color part is called telomeres. So this is a short from the centromere is short is called the P arm, and the longer arm is also known as a Q arm. These two type of the arms are present in the chromosome. Structurally, each chromosome is differentiated into the three parts. It is also completed by it is a pellicle, matrix, chromosomes. It is also double L. It is also last slides are the two chromosomes are also tightly coiled. Then together that they appear as a single thread of the about 800 angstrom thickness. A metaphase chromosomes the following structural features, except chromomers, and we have been under the light microscopes. So this light microscope, 7 parts are there here. First part is also chromatid. Second one is a chromonema. Third one, chromomers. Fourth one, centromere. Fifth one, secondary construction or nucleolar organization. Sixth one, telomere. And seventh one is the satellite chromosomes. These 7 parts are easily and conveniently are seen on the electron microscopes under the during the mitotic phase. So this is also what is the means of the centromere. Centro means center part. The centromere, a small structures in the chromosome marked by a constructions which is recognized the permanent structure of the chromosome is termed as a centromere. Centromere is a permanent, it cannot changes, but it is also called attached part of the two chromatid parts. So at the point, the two chromatids are joined together. What is a centromere? The centromere is a two chromatid jointed part. So it is also known as a centromers, also called a kinetochore or primary constructions. So in division divides the chromosomes into the two sections or arms. Two sections means upper arm, also called a lower arm. Then upper arm is also one chromatid, though lower arm is also other chromatid. The two chromatid jointed part is called centromere, also called a kinetochore. The chromosome is labeled the P arm and Q arm. So then it's a position of the consists of a given type of the chromosomes and form a features of the identification. The identification of the centromere here, the centromere this black color, also called a coloring part. Then the white color part is also called a nucleotide or molecules. Here is a terminal part, the red color, the telomeres. Here is also telomeres elongated and short chromatid and elongated chromatid, two parts are there. But the secondary constructions are nucleolar organizers. So then the chromosomes besides which are having the primary construction or the centromeres passes secondary constructions at any point of the chromosomes. Primary also secondary. First formation, second formation of the chromatin parts. A constant in their position and extent these constructions are useful in identifying a particular chromosomes in effect. So the chromosome region distal to the secondary constructions. The primary and the secondary construction. Example, the region between the secondary construction and the nearest telomeres is known as a satellite. Secondary constructions are near to the telomeres. Telomere means terminal part of the chromosome, that is called a satellite part. Satellite is also a special part of the chromosomes. Therefore, the chromosomes having a secondary constructions are called satellite chromosomes or SAT chromosome. Secondary construction, primary construction between the two chromatin jointed parties centromere, but also called a secondary construction the same way. Therefore, secondary construction are also called the nucleolus organizer regions. Nuclear NOR means nuclear organizer regions, and SAT chromosomes are often referred to as a nucleolus organizers, also called chromosomes. But here is also chromosome organizations. These chromosome organization is important of the secondary construction and primary constructions are there. But primary and secondary, but in the terminal part is called a telomere. These are specialized ends of a chromosomes which exhibit a physiological differentiation and put a polarity. Then physiologically also got differentiated in the primary and secondary constructions. The differentiation and polarity, each extremely of the chromosome due to its polarity prevent other chromosomes. Each chromosomes are prevented to the other chromosomes. That is the segments from being fused with it. The chromosomal ends are now as the telomeres. The chromosome end parting or last part of the chromosomes, which is also telo. Telo means here term or last or ending part. If a chromosomes are breaks, the broken end can fuse due to the lack of telomeres. Broken part is attaches. The broken part, which part of the other chromosome is broken, that part is attachment is by the telomeres. These telomeres are a important fusion, important character to fuse due to the lack of telomeres, due to the lack of the two. Then types of the chromosomes. How many type of the type? The totally how many group of the cells in a plants or animals? Two groups. First one vegetative group, second one reproductive cell. In the same way, the first vegetative group cells having a chromosome is called is autosomes. Then reproductive cells, which is examples of a pollen grains. The also called egg also ova, which have consisting of the chromosomes is called a sex chromosomes, also called a allosomes. Autosomes, allosomes are also called as sex chromosomes. Now, human chromosomes are two types, autosomes and sex chromosomes. Genetic traits that are linked to the sex of the persons are passed on through the sex chromosome. The rest of the genetic information is present in the autosomes. Only genetic informations are carrying the part of the cells is called a genetic, means also reproductive cells. Then only vegetative characters, which is called autosomes. These are two types. Then the humans have 23 pairs of the chromosomes. In other cells of the which 22 pairs of autosomes and one pair of the sex chromosomes in human beings. The chromosome making a total of the 46 means 23 pairs. Then the 23 pairs are 46 chromosomes each of cell. So on the basis of the number centromere, monocentric. how many number of the centromeres are present in each chromosome. Only one centromere present, that is called a monocentric with one centromere. Then also dicentric with two centromeres. Then after di, we also called a poly. Poly means more than two. More than two centromeres are present in the chromosomes. That is called a polycentric chromosomes. So it is the eccentric without centromeres also there. That the four type of the centromere base to the formation of the chromosomes. First one is without nucleus, without centromere is called eccentric. Only one centromere, monocentric, two centromeres, dicentric, more than two is called a polycentric types. Then the polycentric or broken segment are of chromosomes that do not survive for long time. This is so, but here is the chromosomes are divided into the two pairs, P and Q arms. P means short arm and elongated arm, long arm. The two types are there here by either side of the centromere. So which with a construction point called the centromere in the middle part. So this is also construction parts. Here is a derivative. The chromosomes are divided into the two parts, P and Q arms, with a construction point called the centromere in the middle part. The centromere is a coloring substance which either side of the two parts is called a chromatids. The centromere then arrange the centromere arranges between the centromere to either side of the chromatid nature. The chromosomes are which divide the four types: metacentric, submetacentric, acrocentric, telocentric. Then metacentric means the centromere is the middle, meaning P and Q arms. P means short, then Q is elongated arm. The two arms either between part, that is called centromere, which is also called a metacentric. The V-shaped chromosomes are there. Then compare the length. Example, chromosomes are 1, 3, 16, 19, 20 of human beings, which is also called a metacentric chromosomes. Meta means equal. Either side of the centromere, two equal of the arms, also called a P arm and Q arm. P arm and Q. Then submetacentric. Then centromere of center means not a one is also elongated, part P is also elongated, and L is also called just small. Both are called which arranges to the centromere part, that is called it is metacentric, submetacentric chromosomes. The centromeres are off center, leading to the shorter P and are related to the Q arm long. Example of the human beings: 2 second chromosome, 4th, 12, 17, 18, which are all is also called the submetacentric chromosomes. Next one is acrocentric. Acro means the centromeres are severely offset from center, leading to the much shorter. First is the first part of the chromatid is a much longer, and other is also got a much shorter, more difference. That is called a acrocentric. Examples of the chromosomes in human beings is also called a 13 pair of the chromosome, 15, 21, 22, and Y. Y chromosome is also XY chromosomes which are present, which are called the male. The male on the Y chromosome is also, that is examples of submetacentric. Next one, acrocentric. The centromere is a severally offset from the center, leading to the much shorter than the other chromosomes, other chromatids. Only much shorter, but it's called a submetacentric or acrocentric here there. So this is a acrocentric part. This part is a centromere center part. Then either side is a more difference of two chromatids. Then acrocentric. Then telocentric is a, here is a telocentric. Then the centromere found at the end of the chromosomes, meaning so no arm in the existence. Chromosomes are not found in human beings. So this type of the chromatids, this type of the chromosome structure is a not in human beings, which is only the based the human beings metacentric, submetacentric, and acrocentric. Then telocentric is absent. but other organism also called telocentric is present in the same way. So then few organisms of the plants or animals, the four type of the chromosomes are identified based on the arrangements between the two chromatids of P and Q. P and Q type of the chromosome. Then functions and significance of the chromosome. What are the function? Function means it is activity. The number of chromosomes is constant for a particular species. One species to other species, the number is also called a constant. Therefore, these are of a great importance in the determination of phylogeny and taxonomy of the species. Phylogeny means the phyllo means evolutionary characters are the carries or changes. Phylogenic means that is also then and taxonomic. Phylogenic taxonomy means that a phylogenetic classification like Engler and Prandtl, Takhtajan, Cronquist. That is phylogenetic classification means the arrangement of the plants and animals development is the most primitive and also advanced type. So these primitive plants are how to develop the advanced type of the plants that within the during the large number of the periods or more number of the periods. So these primitive organisms are developed into the advanced character. That type of the studies is called a phylogeny. So the genetic code storage. The second point is the first point is the number of chromosomes is a constant. Second one is the genetic code storage. The chromosomes are contains the genetic material that is also required by the organisms. The to develop and grow, the DNA molecules are so made up of the chains, also called the units called the genes. The DNA look like chain structures means in the DNA a group of the cells which is responsible to the specific function, specific inheritance function, which carries to the generation to generation. That is called the genes. Which genes and a group of genes. That which are these group of the genes are mixed to form a genome. All groups of the genes is called a genome, nuclear genome. Then also called a mitochondrial genome, then chloroplast genome. The genes are the selection of the DNA that code for specific proteins required by the cell for its proper functioning. Then sex determination. Human beings have 23 pairs of the chromosomes, out of which one pair is the sex chromosomes. You know that XY, XX. The XX is also common feature of the female. XY is also common feature of the male. These only Y chromosomes are which are responsible to the changes of the man qualities or man gender, male or female. So that the sex of the child is determined by the chromosomes passed down by the male. If X chromosome is passed out of XY chromosomes, so the child will be female, and if a Y chromosome is passed to the male, the XX and XY, it is also one qualities of the character features of the chromosomes. That is also one function. So next one, the center of the cell division means the chromosomes are check successful division of cells during the process of mitosis. So this mitosis is also first one here is a the two type of the character is also reproduction process. But here is also cell division means then development. The growth and development of both are called cell divisions. So the chromosomes of the parent cells ensure that the correct formational or information is passed on the daughter cells required by the cell to grow and development correctly. Means the central dogma. The central dogma or control of the cell division, it is also third important function of the chromosomes. Then formation of proteins and storage. Next one. So the chromosomes are direct to the sequences of proteins formed in our body and also maintains in the order of the DNA. So this is also one characters. Then the proteins are also stored in the coiled structures. The chromosomes are in the one coil structures of the chromosomes. So their position and bound to the DNA help in proper package of the DNA. So this one is also one important of the chromosome functions. Then lastly, so what are the history of the chromosomes? Here is what are the means of the history? Huh? Who was invented the chromosomes? What are the differentiation of the chromosomes? Here is in the based on the history, we are prepared the fill in the blanks, which are most important are uses in the PG and also JL, DL degrees. The dash was first used by Walday in 1888 chromosomes. Then the chromosomes were first described by dash Strasberg. Then the chromosomes are best visible during the dash. This is also important of the metaphase. Then the Edward Strasberger was the first to describe the dash. Means angiosperms in gymnosperms and also angiosperms in gymnosperms. The process of dash was accurately described the Strasberger in a plants, means mitosis, which process are to observe under the microscopes by Strasberger. In 1888, it was the discovered that dash undergo meiosis to produce gametes with half of the number of chromosomes is also germ cells. What are the means of germ? Germ means the male and the female reproductive cells. This one. Here is Strasberger established that new nuclei arise only from the dash. Nuclei means it is also called a division. In 1888, it was discovered that dash means germ cells. The demonstration of the dash fertilization in angiosperms was one of the Strasberg key condition, double fertilization. Strasberger was reported the double fertilization is also called. Then also identification of the chromosomes. Then demonstration of the fertilization by double. The Strasberger emphasized that the dash means nucleus. The nucleus plays the center role in heredity during the fertilization. So without nucleus, the chromosomes is not there. But in the nucleus is only karyokinesis response to the two nucleus. In the same way, inside of the nucleus also all is also divides in a similarly or equally to the in the cell division. So that the Strasberger was emphasized that a dash means also nucleus. But here the structure of the chromosomes fill in the blanks. So then each chromosome is made up of dash means DNA. The DNA means spherically structures and a large number of DNA threads which are inside of the chromosomal metafilament. Then the proteins around which DNA coils are called stones. Stones are surrounding the chromosomes. Then stones are rich in amino acids like and like lysine and arginine. It's in the chromosomes local and the histoproteins are histo means it is a one type of the protein which are rich. That is called a two type of the components lysine and arginine. The duplicated chromosomes are held together at the dash centromere. The duplicated chromosomes are called dash means sister chromatid. The point where the spindle fibers starch is called the dash. Kinetochore means central part of the. So then the short arm of the chromosome is called the dash, means P. Large arm is also called a Q. P and Q arms is also joint part is called a centromere. But the long arm is also called a Q. The outer envelope of the chromosome is known as the pellicle. Outer membrane, outer layer of the chromosomes is called a pellicle. The inner substance of the chromosomes are called the dash matrix. Inside of the matrix, outside of the chromonometa, that is called a liquid part, it is called a matrix. The inner substance is called a matrix. The thread-like structure embedded in the matrix is called chromonometa. The total chromosomes which have only three types. Total parts of the chromosomes. Outside of the covering layer is called a pellicle. Inside of the pellicle is called a matrix. And matrix, the freely swimming part is called chromonimeta. Appears as a single thread-like about thickness, means 800 angstrom thickness. It is important. A chromosomes are a consist of a dash, consist of a about dash. 8 microtubules. It is important. So the constructed regions always present out of the chromosome called the dash. So that is called a centromers. Centromers is a centromers also called a kinetochores are which important functions of the chromosomes. So what are the functions? So these functions are the chromosomes are stored dash means genetic information. So the genetic information is important for the storage and transfer of inheritance character. So the second one, the specific segments of DNA that codes for proteins are called the dash genes. So these are genes are important parts are there. So then chromosomes are play a vital role in the sex determination. Then in humans, the presence of a dash Y chromosomes are determined the male. The chromosomes ensure accurate dash means transmission. Then the process of the ensure equal distribution of the chromosome is called the mitosis. So the chromosomes are regulate the dash means synthesis of proteins in the cell. Synthesis of the chromosomes help maintains the dash of DNA means it is organization. Organization. The woman, the number of chromosomes in is dash is a given species is constant. The one chromosome number to other chromosome number of the plants are most different, not in a same. The chromosomes are helping dash means are in the plants taxonomy means identical characters. So which characters are cell based to identification of the plant name in phylogenetic classification. The phylogenetic classification is it is a well-developed type of the classification. It is also evolutionary type of classification. The chromosomes are helping dash means also taxonomical level. But here is a important of the chromosomes which is also divides the two type euchromatin and also heterochromatin. What are the euchromatin and heterochromatin? Just 5 minutes. So what are the U and also hetero? What are the differentiation? Means the chromated parts of the chromosome based on the storage of genetic information. The parts of the chromatid divides the two. First one is a euchromatin, second one is a heterochromatin. Then loosely packed, less condensed. Less condensed part euchromatin. Tightly packed, highly condensed is called heterochromatin. Transcriptionally active genes can be transcripted into the euchromatin type of the part. Then the transcriptionally inactive genes are generally not transcripted. In the plants, the animals which have two types are there present. Then the staining likely stain under the microscopes. The microscopes are which observe then the thick color of the chromosomes and a light color of the chromosome. Two parts are based on the staining process. Staining. Also darkly stained under a microscope. Location found in the eukaryotic region of the chromosomes. Euchromatin which are present in the eukaryotic. Then also heterochromatin found in the heterochromatin region of chromosomes, but less repetitive DNA, rich in a genetical GC content. That is the molecular organization. Guanine cytosine is also called in the rich content. That is called euchromatin. Highly repetitive DNA, rich in GC content, guanine cytosine content. Then genes actively transcripted during the cell division. Example, transcripted. Transcripted means the genetic characters are transferred to the other organisms. That is called transcripted. Then here is also functions are it can be found in the arms. staining after function. The DNA components contains the repetitive DNA rich in a kern. Then histone modifications associated with a acetylation of histones means gene actions. Here is a histone modification process. But here in the heterochromatin, so this is also associated with a methylations of a stones. That is also important. Microscopic appearance means it appears in the disappears and highly condensed the chromatin appears and disappears. It is also highly condensed part. That is a microscope. It can appear in the dense and darkly centered chromatid. Examples: actively transcripted genes during the interphase. In the interphase, which chromosomes are more active, that chromosomes are examples of a euchromatin. But here is appears to the dense and darkly stained chromosomes. So this is also one character example. DNA in the acceptability, high open configurations allows the binding transcription factors. The euchromatin have high type of the chromatin parts. Then heterochromatin low closed configuration. Then recombination activity high in euchromatin, high genetic activity crossing over meiosis. So that chromosomes have, which chromosomes has a more percentage of the euchromatin, it can be ready to the mutations and evaluations are there. High open configuration binding process, but recombination process is high in euchromatin, that is also heterochromatin low. Then presence of the cell cycle prominent during the interphase and active genes are present. Here, that euchromatin, but here heterochromatin low suppressed recommendations. Presence of the cell cycle, it is prominent during the interphase and active genes also expression. Then the heterochromatin is also remain condensed throughout the cell cycle. Then also called enzymatic sensitivity. So this enzymatic sensitivity to DNA open structures. Here, resistance of DNA closed structures. Role of the gene regulation in both are called actively role in expression function is called eukaryotic. Then silence of the down regulating the non-essential reports parts is called a dead parties of the chromosomes. Then replication time early in S phase, but later in S phase. So then evolutionary role, the namely solving a regions or evolving a regions. We also concerned the table as table of DNA regions. So this is also made differentiation. So it is also complete of the what are the differentiation of the euchromatin and heterochromatin. How many important bits which can ask to the GL and also called intermediate, GL and GL PG entrance coaching, also DL entrance coaching, or is also here. That is called a chromosome structure only, not the chromosome chemical composition and also complete structure of the heterochromatin and euchromatin. It can be takes in the next class. Okay, sir.
Sir, today's session is completed, sir.
Yes, sir. Completed.
Okay, sir.